Hello, everyone. My name is Boris Aguilar. I work in the Institute for Systems Biology in Seattle. And today I wanted to talk about a work in which we are using imaging features together with machine learning methods to try to predict recurrent events and subtypes in uh, white and black patients of breast cancer. And our goal is to try to identify markers or mechanisms that can explain the disparity in aggressiveness in breast cancer between these two groups. And we focus on these two groups because it has been observed before that for young patients of breast cancer, black patients have a more aggressive breast cancer than white patients. And that figure shows the survival curve for this and clearly shows the differences between these two groups. One of the properties that we are focused on is subtypes or molecular subtypes. And this is important because it has been shown that the incidence of basal-like subtype is higher in black patients. And this subtype, basal-like subtype, is one of the most aggressive cancers or breast cancers compared to the other subtypes. And these subtypes were identified using PAM50 algorithm from gene expression data. But when we also use other types of subtype classification based on immunochemistry, it has also been shown that the incidence of triple negative breast cancer is higher in black patients compared to other races. And importantly, many patients that are triple negative are also basal-like subtypes. So that's another indication that there is a disparate aggressiveness when we compare black participants with white participants. And the other property that we are trying to predict is recurrence in breast cancer, which is when you have a breast cancer that comes back after treatment, and it can be local recurrence, regional recurrence, or distant recurrence. Distant recurrence is metastasis. And it has been also shown that the black race was significantly higher, have associated with significantly higher distant recurrence rate in some special subtype of breast cancer in black patients. So given these previous observations about disparities in these two races or groups, our goal was to characterize these disparities using imaging data or radiomic data together with machine learning. And we will be trying to predict subtypes and recurrence. And our goal is to identify potential markers or potential properties that can explain these disparities. For the analysis, we are using a data set that was published together with this paper. This data set is a comprehensive data set that contains 922 participants, and it has radiomic features, which are numerical or quantitative properties derived from medical imaging, like MRI imaging. It also contains demographic and clinical data, treatment outcomes, and molecular subtype that is needed for our analysis. This figure here is trying to show our methods from the original data set that contains 922. We considered only participants with our Black or African American and also white, and 50 years or younger. At the end, we end up with a data set with 346 participants, from which we collected imaging data. So this data set contains 529 imaging properties, and they are organized in 10 groups or 10 categories. And that's our imaging data. But we also use molecular data. We collected gene expression data from this data set called TCGA. So with radiomic data and molecular data, we used machine learning techniques to predict recurrence and subtypes, but we also use information gain as implemented in Boruta to identify key molecular and imaging features in these two races or groups. And all our data was transformed and in tables, and they are now in the cloud. Specifically, we used Google BigQuery. And then from that, we used all of the resources of the cloud to do some statistical analysis. Specifically, we used Wilkinson test to identify associations between these numerical features and race. So this table shows our results when we try to predict molecular subtypes. The numbers are F measures that tells you about how accurate we can predict. And we used three machine learning methods to try to predict one of the four molecular subtypes. The original data set is unbalanced, meaning that there are differences in population between these groups, but we also use SMOD to balance this data set. And we did 3 experiments. In the first, we used imaging data only to try to predict subtype. Then we use gene expression or molecular only to try to predict subtype. And finally, we try to use both, combining imaging and molecular subtypes. So the numbers here are telling you that we are good at predicting subtypes using these data sets. But one observation was that we really need to balance the data set to be able to get better predictions. And the second conclusion from this is that when we combine imaging data and molecular data, we get better performance in predicting subtype. And here is a list of features ranked in importance to predict subtype. And one observation was that the most important features are the molecular features, that are genes. And the most important imaging feature is at place 15. So the conclusion is that the most important feature to predict subtypes are gene expression of these patients. This plot is trying to show you the same. In the y-axis is the importance scores, and the x-axis are the features. There was few space to put the names, but they are the features, and you see that the most important feature is this gene, and the most important imaging feature is below in place 15. So molecular features, gene expression features are more important to predict subtypes. This table here is showing you the results when we try to predict recurrence. And here we only use imaging data to try to predict recurrence. And these imaging properties are organized in 10 categories, which are the rows in this table. So we try to predict if the patient presented a recurrence or not. Again, these numbers are F measures, and we use it to implement two machine learning methods, random forest and AdaBoost. And then we also did 3 experiments when we tried to use all the patients, and we tried to use only white patients and only black patients. And as you can see, the methods can predict recurrence with a good level of accuracy. And we didn't observe differences in prediction capacity between white and black participants. And this table here is showing imaging features that are statistically associated with race. And this is the p-value. In this feature, if you compute the average in black patients, we found that these are different from white patients. That can be used later to try to explain the differences between these two groups. And in this table, even though we observed similar predictions when we use black patients or white patients, the most predictive feature for white patients is different than from white patients. For example, here in this category of imaging features, breast volume is the most predictive feature for black patients. But for white patients, we found it's a different one. It is tissue volume post-cone. Importantly, these features are significantly different when we compare black and white patients. So there are many more categories in which the most important or most predictive feature is different between these two groups. But there are some groups in which they have the same most predictive feature. And this plot is showing you for one of this category, for TESH, I think it's this category here, tumor enhanced spatial heterogeneity. This plot is showing you the importance scores for the different features in this category. Thank you. And we found that in this, for black patients, this is the most important category. And if we do the same for white patients, the most important feature is different. And another observation is that these features have a very high score for white patients, but a lower score for black participants. Okay, so as a conclusion, we analyzed the disparity observed in these two groups, and we characterize differences using molecular data and also imaging data, and try to combine them both to make predictions in recurrence and subtypes. We used several machine learning techniques to predict recurrence at subtype, and our main conclusion from this preliminary work is that we really observed that when you combine imaging with molecular subtype, you have a better capacity to predict recurrence and subtype. And even though when we tried black patients and white patients and observed similar predictive capacity, we observed that the most predictive feature, imaging feature for black patients is different than from white patients. And that can be used later in future work to try to understand why we observed these disparities when we compare these two groups. Finally, I want to thank our collaborators, George Aquamensa, who is here, Kauter Advile, and Ronald Taylor, who is also here, for their work. We work this together. And then funding and support, I want to thank ISB CGC, which is ISB Cancer Gateway in the Cloud. They provide us resources and credit to use the cloud for this analysis, and also NIH for funding of my work specifically. All right, thank you for your attention.
Thank you for your great talk. I'm not very familiar with radiomic features. Is there like some sort of biological meaning behind those features, or what do you think those features indicate that are predictive of recurrence that differ between black and white individuals?
These features, there are like 500 of these features, and many of these are related to the geometry of the tumor, like tumor volume, or how they spread, the intensity of when you look the images, And so it's possible that the morphology of the tumors can tell you about the aggressiveness of cancer.
Nice, thank you.
Yeah.
I just have one small question. So usually there is a delay in diagnosis depending on the sociocultural demographic of the person. So I was wondering if at some point the triple negative worst cases are related as well to the delay in diagnosis, and then that's what you see as an ethnicity relation.
Yeah, it is possible. I think we were constrained with the data set that we use it, and I guess in the future we can explore larger data sets and include these properties in our analysis. So far, we didn't analyze those.